Dear viewers, welcome to my YouTube channel. Today we are discussing about biosynthesis of phospholipids. I am Satyaraj from MSc Applied Microbiology, Department of Microbiology, Sacred Heart College, Tirupattur. Phospholipids definition. These are compounds containing, in addition to fatty acid and glycerol, a phosphoric acid, nitrogenous bases, and other substituent. Polar compounds composed of alcohol attached by phosphodiester bridge to either diacyl glycerol or sphingosine. Amphipathic in nature as a hydrophilic aid, phosphate plus alcohol. Example serine, ethylamine, and choline, and a long hydrophilic tail, fatty acids or derivatives. Classification of phospholipids. Phospholipids are classified according to backbone content into glycerophospholipids and sphingospholipids. Glycerophospholipids is a backbone glycerophospholipids composed of glycerol, two fatty acid, phosphate group, and alcohol amine. Sphingospholipids backbone is sphingosine. Sphingospholipids composed of sphingosine, alcohol plus one fatty acid plus phosphate plus choline. Both classes are found in membranes and play a role in generation of lipid signaling molecules. Glycerophospholipids are formed by phosphoric acid and an alcohol. It is a formation of phosphatidylserine and phosphatidylethanolamine, phosphatidylcholine, phosphatidyllinositol, phosphatidylglycerol. Sphingospholipids are sphingomyelin. Backbone of sphingomyelin is the amino alcohol sphingosine rather than glycerol. A long chain fatty acid is attached to the amino group of sphingosine through an amide linkage, producing a ceramide which serves as a precursor of glycolipids. Sphingomyelin is an important component of myelin and nerve fibers that insulates protects neuronal fibers of the central nervous system. Synthesis of phospholipids. Biosynthetic process. One of the hydroxyl of the phospholipid is first activated by attachment of nucleotide cytidine diphosphate. Cytidine diphosphate CDP. It's also called CDP, cytidine diphosphate. That CTP attached either to diacylglycerol forming an activated phosphatidate CDP diacylglycerol. Strategy 1 and strategy 2. In both cases, CDP bound structure considered an activated intermediate and CMP is released as a side product of GP synthesis, glycolysis process synthesis. A key concept in phosphoglyceride synthesis is activation either diacylglycerol or alcohol to be added by linkage with CDP. It is a structure or pathway of CDP. Synthesis of P and PC from pre-existing choline and ethanolamine. It is a synthetic pathway involves in phosphorylation of choline and ethylamine by kinase followed by conversion to activated form CDP choline and CDP ethanolamine. It is a pathway of CDP choline pathway. Choline, phosphocholine, CDP choline, phosphatidylcholine. Synthesis of phosphatidylethanolamine and phosphatidylcholine. PC and P are most abundant phospholipids in eukaryotic cells. The liver requires mechanism for producing PC even when free choline levels are low. The provide needed PC and PS is decarboxylated to phosphatidylethanolamine by PS decarboxylase. P then undergoes three methylation steps into produce PC. It is a major pathway of eukaryotes. Phosphatidyl serine. Primary pathway for synthesis of phosphatidyl serine in mammalian tissue is provided by the base exchange reaction in which the ethanolamine of P is exchanged for free serine. This reaction, although reversible, is used primarily to produce and P is required for membrane synthesis. Phosphatidyl linositol. It is synthesized from free inositol and CDP diacylglycerol. It is a pathway of CDP diacylglycerol. Sphingophospholipid. Sphingosine is based phospholipid is major structural lipid in the membranes of nerve tissue. The synthesis of sphingomyelin is shown in figure briefly. This reaction, like the decarboxylation reactions involved amino acid, requires pyridoxal phosphate. Product is reduced on NADPH, requiring a reaction spinganin, which is accelerated at the amino group with one of a variety of long chain fatty acid and then desaturated to produce ceramide, the immediate precursor of sphingomyelin. It is a pathway of sphingomyelin. Functions of phospholipids. It is associated with proteins. Phospholipids from the structural components of membranes and regulate membrane permeability. They participate in the absorption of fats from the intestine. They are essential for synthesis of different lipoproteins and thus participate in transport of lipids. They prevent accumulation of fats in liver. They act as a surfactants. Cephalin participate in blood clotting. Phosphatidyl inositol is a source of second messenger that are involved in the action of some hormones. Role of sphingolipids. Abundant component myelin sheath, also widely distributed in membrane of plants eukaryotes. These lipids are continuous state of turnover. Defects in this metabolism leads to various disease. Thank you.